right, so to help recap where we left off, we started out again just like we did in lab, differentiating a skeletal muscle from that of a skeletal muscle cell or fiber. And now we need to turn our attention to an actual individual muscle cell or muscle fiber, because as we learned in our lab last week, skeletal muscle cells have some really unique properties about them. So let's go back and review these properties. The first thing is we know which type of nerve controls our skeletal muscle. It is our somatic nerves, which gives us what type of control? Voluntary. Okay. It does control the body. It gives us voluntary control over our skeletal muscles. We also said our skeletal muscle cells have the membrane can have a unique name. What's the unique name of our skeletal muscle cell membrane? Sarcolemma. Sarcolemma is a name for our skeletal muscle membrane. We just know that the prefix sarco is just talking about skeletal muscles, right? And the sarco translates into flesh. We also learned that the sarcolemma can project inside of the cell, and when it does that, what do we call those projections? The projections of the sarcolemma into the cell are called T-tubules, or transverse tubules. And I may or may not have noted in class on Tuesday, on these big images that you have in your guided notes, there should be plenty of room to add not only just the labeling, but any details. So this will be your note-taking area. So our sarcolemma does project into the muscle cell in these things called T-tubules. The T stands for transverse tubules. What did we say the function of the transverse tubules are? Conduct action potentials. So our transverse tubules can conduct action potentials. Just like so can our, what else of the muscle cell can conduct action potentials? Conduct them. It actually is responsive to them. It doesn't do the conducting. The sarcomeres, yeah. Sarcomeres do not conduct action potentials. Those are the part where the cell will do what? Just the sarcolemma. The muscle cell membrane conducts action potentials. Sarcolemma conducts action potentials. So all that background information that took us a couple of weeks to understand in the nervous system also happened here at the sarcolemma. But instead of me going back to September and saying the pump is pumping, the leaky channels are leaking, there's a gradient, there's voltage gated channels, right? Instead of doing all that, we're going to imply all those factors are here in the sarcolemma. And so our muscle cell membrane can maintain a resting membrane potential. And you can also conduct action potentials. So can the T-tubules can conduct action potentials. The transverse tubules are adjacent to another organelle called what? The endoplasmic, or you can call it the sarcoplasmic reticulum. What color is it in this image? The purple, blue, right, something. The sarcoplasmic reticulum is a specialized what? Not rough, smooth ER. The sarcoplasmic reticulum is a specialized smooth ER. Specialized for what? Releasing calcium. Storing calcium and releasing calcium. So our sarcoplasmic reticulum then, in order for it to release calcium, should have what about it? Right. Our calcium channels, what kind of gate should be on them though? Correct. The voltage gated channels are expressed in our sarcoplasmic reticulum. So we said it's sensitive to the action potential that is conducted by the adjacent transverse tubules. Also shown in here is a high abundance of mitochondria. What can we take away from that then? If a muscle cell has a bunch of mitochondria, what can we take away? It needs a lot of ATP, and it can make it because it has a high amount of mitochondria. So our muscle cells do rely a lot upon aerobic respiration. Krebs cycle, lipolysis first, Krebs cycle, and the electron transcription, the power muscle mechanics. I think we may have briefly mentioned on Tuesday, but I'll go ahead and say it again just in case. Your skeletal muscles are multi-nucleated. What does that mean? They have a lot of nuclei, right? Several nuclei within a single fiber or a single cell. Your muscle fibers, your muscle cells, are multi-nucleated. This is due to the way that our muscle cells grow and develop, is that individual cells fuse together and become one. And then so multiple cells will grow together and make a single continuous muscle fiber or muscle cell. Additionally, each of our muscle cells are packed with things like we can see stretched out of here. What is that representing? Thing being stretched out. What is that? What is it? Call it a myofibril. Myofibrils give the cell the ability to contract. And these myofibrils are arranged in repeating units called what? Sarcomeres. We're going to review or introduce sarcomere structure today. We'll talk about its features. The myofibrils are just repeating units of these sarcomeres. And so to understand how a myofibril contracts, we really need to look at how a sarcomere is built and how it contracts. We know how a sarcomere contracts. We know how a myofibril contracts. And then we know how an entire cell can actually contract. The number of myofibrils is variable in different tissues, right, or different cells. Cells that need more strength would have more myofibrils. Cells that need less power ability would have less. Or let's say you just don't use a muscle that maybe normally would have the need for a lot of powerful contraction. What's going to happen to the myofibril count? It's going to decline. I've heard of muscle atrophy. So the myofibril content will decrease. You probably didn't lose any muscle cells, but you lost the myofibril count in those cells. Well, if you exercise more, your myofibril count has gone up.
What were you saying? If you lose them, you can't get them back. What aspect does that have to do with?
If you lose a muscle cell, it's not going to have the ability to regenerate. If you lose a myofibril within a living, surviving cell, it can be regained with exercise, or just use of the muscle. And so the myofibril content can increase or decrease based upon the use. So there's no reason for, if a muscle cell is not being used, there's no reason for your body to spend energy just maintaining something, so it can catabolize it and break it down. And that's why you quickly see disuse atrophy if someone gets a limb and a cast or whatever the case may be. Myofibril counts go down, but then as the activity goes up, we can build more myofibrils back in there. Good. Additional questions or clarification? I want to turn our attention now to that kind of panel over here in the window panel. What is this area illustrating? Neuromuscular junction. That is illustrating what we were going to call a neuromuscular junction. That relationship between a somatic nerve and a skeletal muscle cell. We know that our somatic nerves release acetylcholine. We know that our muscle cells have which receptors? Nicotinic receptors. So go ahead and on your motor end plate of your muscle, draw some nicotinic receptors. Just some chemically gated channels for which ion? Sodium. So on your motor end plate, draw some nicotinic receptors. Where's the motor end plate? The y'all are doing this, okay, portion under the nerve, right? So this part of the muscle cell that actually has the nicotinic receptors. You want it where the acetylcholine's released. So it's just right underlying the somatic nerve. And in this image, it's highlighted or bolded a little bit. So add in some nicotinic receptors there. These are, what type of gated sodium channels? They're not voltage. What are they open to? Chemical. They open in the presence of acetylcholine. They open in the presence of acetylcholine, a chemical. So these are what kind? Chemically gated sodium channels. Would they open in response to voltage? Would our nicotinic receptors open in response to voltage? No. They're not built to respond to voltage. So again, what type of receptors are located here? Nicotinic, they are chemically gated sodium channels. When opened, sodium will flow in or out? Into the cell, just as it would in a nerve cell. So we have the sodium-potassium pump pumping sodium. Sodium-potassium pump sodium. My head's going to do this right now. Sodium-potassium pumps 3 sodium out and returns 2 potassium in. It's working just the same in your muscle as it would in your nerve cell. And so we have sodium at rest in greater concentration outside of the cell. So if I open up a channel, sodium will flow in. All right, happy dance. Sodium will flow into the muscle cell, causing it to depolarize. Does acetylcholine go into the muscle cell?
No.
It doesn't. It's just they're like the key to open the door. The door lets it lets mostly sodium, which way? Into the cell, down its chemical gradient. These are located where? Under the neuron, we could say at the motor end plate. What's the green thing next to that motor end plate? Transverse tubules. Will it have any nicotinic receptors on it? No. It's not anywhere close to the somatic nerve, really. What is on there, though? There is some receptors. Your book goes into the names of them, but we haven't used their names. So you're right. What are they? What is in the membrane of our transverse tubules? We're going to have different types of voltage-gated channels. So why don't you draw a voltage-gated channel in your T-tubules? Again, these features are outlined for you in figure 12.2. Before we move on to look at the sarcomere, what questions do you have about some skeletal muscle cell features. All right, if some questions come up later, just ask me later. I'm going to turn our attention now to a sarcomere, and I may not have given you enough room on the sarcomere structure. Yesterday's class got along okay with fitting everything in, so what we're going to do is look at a sarcomere. These things make up our what? Myofibrils. M-Y-O-fibrils. Myofibrils. The myofibrils are just basically protein rods of these repeating units called sarcomeres. It's these sarcomeres that give the striated appearance to our, not only our skeletal muscle, but also to our cardiac muscle. So if you look at a long section of a skeletal muscle, you're going to see this repeated banding, light, dark, light, dark. And hence the striation appearance of the muscle. On the top is a cartoonized kind of image from your textbook. And the image on the bottom is a real transmission electron microscope image of a sarcomere. A sarcomere extends from what's called Z-line to Z-line. You can call them Z-band, Z-line, Z-band, Z-disc, whatever, but the letter Z is what you're going to see the preceding Z. I think it's the same, or the nomenclature, because it looks zigzagged. The way the proteins are arranged are zigzagged one another. So that's the Z-line. So I have 1, 2, and then a portion of another sarcomere. So we're looking really super zoomed or magnified in this case. In the transmission image, the Z-line is shown as this kind of darker looking band. If you look at the Z-line, not only is it zigzagged, it's also, what it's serving as is a point of attachment. Does anybody know what we're attaching here? Actin, which is part of our thin filament. Okay, your Z-line is the point of attachment for your thin filaments, shown here in the purple. Again, the Z-band is zigzagged, but it's also functioning as the point of attachment for our thin filaments. The very middle of the sarcomere is called the what? Even more central than the H-band, something called the mline or the midline. Okay, is right in the middle. So this is this protein running perpendicular to our thick and thin filaments. This here, the mline, shown right here. Have another one shown here, and another one shown here. If you look in the TEM image, you notice real closely, can you see this light area? That is the mline. Looking even, so yes, it's anatomically in the middle of the sarcomere, but it also serves as an attachment for what? What is attachment? The thick filaments. The mline is our point of attachment for thick filaments. Z-line attaches the thin filament. Thin filaments are attached to the Z-line. Thick filaments are attached to the M-line. And you notice that thin and thick filaments are running kind of parallel with one another, and they do overlap some. And this overlapping, or lack thereof, is what gives us this dark and lightness. So if you're looking at something through a microscope, with a light microscope, you're trying to see how much light goes through a sample. If there's a lot of proteins or other cell features in an area, could more or less light go through? Less, and so the area would look darker. In the areas where there's less of something, more light could go through, and so it'll look lighter. And that's what's happening here in the sarcomere. We can see the light and dark, just the way that where there's more or less proteins, right, concentrated. analogous feature is in a transmission. Instead of using light, electrons are used. And so thin samples, electrons can pass through. Thicker samples, electrons bounce off. And so they still have this light dark appearance when the image is processed. So again, in this image, the thick filaments are colored as the orangey red barbed things. The thin filaments are the purple straight lines. We'll pick up thin and thick filament structure after we just look at the orientation in the sarcomere. In addition to the mline and the zline, we have different bands and zones. We're going to talk about the iband, the aband, and the H zone. The iband is referred to as the light band, so using the first vowel, or the only vowel, light band. And the iband looks lighter than the dark band. The light band is light, the dark band is dark, so the iband is the light band. It's the part of the sarcomere where the zline is, where there's only which filament? Thin filament only. No thick filament. So if I were to look at the iband, if I look at the zline and then just adjacent to it, it looks lighter. Okay, so that's going to be the iband. And I'll highlight it here in this image with the purple. So that's going to be your iband. Where else can I draw an iband? There are two more, yes, two more. So right here, I could put another one, and right here, I could put another iband. What's in between those? The aband, the dark line, which would have the mline in it, but more broadly, there would be the aband. The aband is any part of the sarcomere that has what in it? The thick filaments. Here it is going to be myosin. So anywhere there's thick filament projecting, it's going to look dark. And we call it the aband. And it's highlighted here in the green. So we're going to go light, dark, and we can keep going. However, if you scrutinize even more, if you notice right in the middle of the aband, there's a less light area of the dark area. Can you see that? It's not as dark as the rest. Right around the mline, where our thick filaments attach, and there are no thin filaments projecting, it looks a little bit less dark. What is this called? The H zone. I don't know where that came from. I don't know if it translates to something, but it's right in the middle. It's called the H zone, highlighted here by the blue box. So the H zone is in the middle of the what? The aband, and it's the part where there is no thin filament. Uh-huh. If the sarcomere is built right, she's right in the middle.
Purple boxes are the ibands and the green boxes are the abands on the top.
Yes.
Okay.
The purple boxes are the ibands. The green boxes are the abands. And in the middle of the abands is a less dense area called the H zone, is highlighted by the single blue box shown ahead. If I were to do it here, I would mark it right here. H zone.
And it's where there's no thin.
Correct. It is, the H zone is part of the dark band, but there's just exclusively thick filaments. There are no thin filaments overlapping. So even though there's thick filaments, electrons can get through there easier than they could where there's thin and thick filaments overlapping.
So the A zone everywhere but the H, or the A band everywhere but the H is both thick and thin?
Yes.
Okay.
Yes. So if you look on the other sides, the A band would have thick filaments projecting this way, and then thin filaments remaining this way. And they'll just physically overlap one another. We can do a very simple analogy, it seems to work. So I want you to take your sarcomeres, okay? And your thumbs are going to be your Z-lines. And just take them and overlap your thin and thick filaments. In the relaxed state, tips kind of overlapping a little bit, and then contract. And I want you to notice how the light, say your iband, becomes less prominent. So during contraction, we can change the iband shape, and then we're also going to see the H zone is going to shorten as well. But this is what happens when your sarcomeres overlap. If they don't, the thin thick filaments don't wrinkle or crinkle or bend or whatever, they just overlap one another. And so, for example, the length of the thin thick filaments, like the length of your fingers, don't change, but the distance between the Z lines, your thumbs, does change. And it's not real profound, but it's enough to promote shortening. What other questions do you have over the sarcomere structure before we turn our attention to the thin and thick filaments? Z-line, M-line, I-band, A-band, H-zone. Comfortable? These represent a single what? Parts of a single sarcomere, which bind together to form a single fibril. If you have multiples of these, you could find them packed into a single what? Muscle cell or muscle fiber. So we use the word thin and thick filament pretty easily, I think, but we really need to understand the proteins which comprise these thin and thick filaments. The thin filament gets its name because anatomically its diameter is thinner compared to the diameter of the thick filament. So we're going to zoom in and look at some basic structure. We're not looking at an entire polymerized thin filament. We're not going to look at quite yet an entire polymerized thick filament. We're going to look at the basic components. These are part of our sarcomeres, and these work as contractile proteins, which makes sense because they're used for contraction. These proteins are in families. So when I say actin, we're not talking about all actins are built exactly like this. We're looking at actin in the skeletal muscle of a human. Okay? Actin of a muscle in a fish might be I think a little bit different than mice, but they still work as what? Contractile proteins. Okay. What is the contractile protein of our thin filament? Actin monomers polymerize to form a polymer. Okay. Or you could say actin filaments. So actin, what color is the actin in this image? They're purple glob. The globular proteins shown here, the light purple globular proteins, or blue, whatever color you want to go with. Okay, these guys right here, little bead-looking things. Those are individual actin proteins that adhere together to form a strand, so you call that a fibrous protein. So these things represent actin. Highlighted for you is a dark area. What is that darkened area on the actin molecule? The binding site for myosin. So be sure to label this dark area as the myosin binding site. Each little actin monomer has a myosin binding site. A few of them highlighted. They're dark in your image, and I just highlighted some orange circles so you can see what the heck I'm talking about. These myosin binding sites are actually covered up. What are they covered up by? Tropomyosin. Tropomyosin is this fibrous protein, this thin fiber of protein, which runs along our actin polymer to block the what? The binding sites for myosin. Good. Because these proteins are contractile proteins, so we need to also have our muscles relax. And so we can inhibit that interaction of our contractile proteins by having the presence of tropomyosin. There is a third protein family in a thin filament. What is the third protein? Troponin. Troponin is the third protein in the thin filament. And it's shown here in this kind of lavender. There's 3 little binding sites to it. It has 3 binding sites. What can troponin bind to? It can definitely bind to calcium if you did your reading. We know that it binds to calcium. What else does it bind to? Tropomyosin and actin. So troponin, yes, binds to calcium, but it's also bound to tropomyosin and to actin.
So the calcium binding sites, are they just anywhere on
I don't know where they exactly are on the protein molecule. And reminding you, these little proteins are really kind of simplified for us, but it's going to be more towards the area of the cytoplasm, so that because that's where the calcium is going to come in from. And so it's got to be able to easily bind. And so on your handout, just pick one. It doesn't. As long as you acknowledge it's on troponin, I'm really happy. The calcium binding site. But it'd be more cytosolic facing. You wouldn't want to see it buried under, say, down here, so the calcium wouldn't be able to get to it to access it. So again, let's just review. Troponin can bind to calcium. We said, well, where? It's going to be more on the cytosolic facing portion of it. Calcium's got to bind quickly. But to adhere it to the molecule, it definitely binds to tropomyosin. To keep it all from floating away, it also binds to actin.
So how does the tropomyosin, does it move out of the way from the binding site? How does the
It does. Does anybody know how tropomyosin moves out of the way? Very good. It's a conformational change only when calcium's present. So we go back to putting everything together. When an action potential is present, sarcoplasmic reticulum links calcium. Calcium binds to troponin, and it changes shape. So it's called a conformational change. And so it physically pulls back, and when it changes shape, it's attached to what? Tropomyosin. It's bound to the actin on the bottom, but it pulls back, just like if you're riding on a horse, right? So, and when it pulls back the tropomyosin, what's exposed? The binding sites beneath it.
So that's actually attached, that top one is attached to the tropomyosin, where the bottom one is attached to the actin?
Pretty much. And again, this isn't structurally correct. It's just showing that it has three binding sites. But that reiterates what we just said. It has 3 binding sites. It binds actin, it binds tropomyosin, and it also binds calcium. So when calcium's present, it's anchored basically to actin, and it pulls back tropomyosin. So only when what is present can the thin and thick filaments touch calcium, because it binds to troponin, which pulls back tropomyosin, which exposes the myosin binding sites on actin. So it's just a structure shape. We want to wash it down. Calcium changes its structure and changes its shape to pull back tropomyosin. It's a great question. Only when calcium's present can that troponin pull back tropomyosin to expose the underlying binding sites. Additional questions about the thin filament? This is a portion of the thin filament. It would run along the length of a sarcomere. Okay, so we're just kind of chopping it up and looking at it. If we look at the structure of the thick filament, it's is made up of a molecule called myosin. This is a single myosin molecule which would group together with other myosin molecules to make a big, giant, thick filament. So this is just one tiny part of the thick filament. This image is not an entire thick filament. Same thing, myosins are families, and so they aren't all built exactly the same. We'll see a difference between skeletal muscle myosin as well as from smooth muscle myosin. But they're going to have some similar structures. The first that we can note is that our myosin proteins have what's called a filamentous tail, right, or the tail is straight. And then a head which is described as being globular. Furthermore, a myosin molecule is not one of these, but it's two of them. It's called a dimer, homodimer. It has two of the same proteins interlaced one another. So this is an entire myosin molecule. What does the tail do? What do you think the tail does of this protein? It binds it, what would you say?
Expands?
It actually, I don't think it expands any. It's just there to anchor it in the thick filament. This way we can attach all of our little myosin molecules together. So it's a form of attaching to other myosin molecules to make up our thick filament. And so that's important to hold it in place. But the working machinery that we're going to study is actually at the head of each myosin molecule. And from your handout, you noted that the myosin head has two binding sites. What are the binding sites for myosin? One we better know. We better have a binding site for. Yes, but the one based on our previous discussion, you guys are right, but also actin. If actin has a myosin binding site, myosin probably ought to have an actin binding site. In this, the kind of top portion, that's what's going to have to reach over and touch the thin filament. Mm-hmm. So those would be analogous. Yeah, these would be analogous to receptors. They're going to touch one another. Use the word loosely, but reciprocals of one another. Touch. These are contractile proteins. But as noted, our myosin molecule has to have the ability to bind and split ATP. And so there's an ATPase site on the myosin molecule. So even though this binds and it's a contractile protein, it also has a part of it that acts as a what? An enzyme that splits ATP to extract its what? Energy. Because we're trying to do some type of mechanical work. So we directly just split ATP, take its potential energy, and then power the contraction. Yeah, so on the handout, again, it doesn't necessarily matter to me how anatomically precise you are as long as they're both labeled. There are two heads in your image, on your guided notes, or handout, they're just flip-flopped, okay, just to kind of differentiate them. But each head would have a binding site for each ATP and each actin. So I have two heads, so I would have two separate actin binding sites and two separate ATP binding sites. But it makes logical sense that the actin binding site would be on the edge, right, or the tip. That's what's going to cross over, we're going to use the word cross bridge, over and touch the actin. And energy is transferred, the balance, or the neck, if you will, of the myosin molecule. To answer your question? Yeah. So our myosin molecule is a dimer, meaning two of these proteins, tail that anchors it into the thick filament, in the heads that have the ability to bind actin and also ATP, to split ATP, to take its energy. Your myosin molecules, individual molecules, can polymerize into an actual thick filament. So 12.5C helps visualize this. And that's a single myosin molecule is not going to be the thick filament. We've got to group all these little proteins together, and so you can see how the tails are anchored or adhered to one another to make. Now you can see it's more thick when in comparison to a thin filament. So a single thick filament will have thousands of little myosin cross bridges or heads out there to try to facilitate contraction. What questions do you have at this point about the thick filament? All right, so let's just recap how these thin and thick filaments overlap one another in a sarcomere. Again, our sarcomere is going to extend from Z-line to Z-line. We can see our thin filament shown in the purple, and the thick filaments in the whatever color that, orange or whatever. Not shown is the M-line, so why don't you lightly draw in the M-line? And you know what? You can even quiz yourself really quickly. Go ahead and label the aband, the iband, and the H zone in this image and test yourself. All right, so again, the aband remains unchanged, but the H zone and the ibands become smaller during contraction because the thin filaments overlap more the thick filaments in the contracted state. Which way do the Z lines move? They move towards the M line during contraction, and it will recoil in the relaxed state. Those images are backed up with real transmission images. Figure 12.6b gives you a sarcomere in the relaxed state and contrasts it to a similar electron microscope image with the contracted state. And so the H zone is really not as apparent as it is previously, and the iband also is less apparent. What questions do you have over the sarcomere structure, the overlapping of the thin and thick filaments, or the thin and thick filament structure before we look at how contraction actually happens? We still haven't talked about contraction. We've been talking about muscles for 2 weeks. We still haven't got the contraction yet. Any questions at this point? All right, so let's go back and review the sequence of events. If you turn your attention to figure 12.8, it illustrates what's called the excitation contraction coupling. To back this up, this is a video that I showed in your lab for you guys last week, and then how, in essence, when a skeletal muscle gets excited, is that going to immediately contract? There's a whole series of events which happen that lead up to contraction. So let's go back. We are at a single what? Label this. We are at where are we? At a neuromuscular junction. Here we're at a neuromuscular junction. What type of nerve do we have here? Somatic nerve releasing acetylcholine at an area of the muscle called the motor end plate. The motor end plate. So our somatic nerves release acetylcholine at the motor end plate. Because the motor end plate has nicotinic receptors, which are types of chemically gated sodium channels. So when they open, sodium flows into the cell. So walk through one of your channels again, just draw sodium flowing into the cell. If sodium flows into a cell, the cell will depolarize. So the flux of sodium at the motor end plate causes a graded depolarization. If you read ahead, there's a more concise way to say this. You might know a better way to say this. It's graded depolarization at the motor end plate. Not yet. It's called the end plate potential. An end-plate potential is another way to say graded depolarization at the motor end plate due to the influx of sodium through the nicotinic receptors. Or I could say end-plate potential. That's a little easier, isn't it? That's implying all that background information that we've worked through the last month. An end-plate potential is a graded, what was it, graded depolarization. There's a potential change, it's a graded depolarization at the motor end plate. Conveniently located, though, the rest of the sarcolemma are voltage-gated channels. So what could the rest of the sarcolemma do? Depolarize to threshold and generate an action potential. So on your muscle cell sarcolemma, draw some voltage-gated channels. Those are not shown here. They're implied by these arrows. Draw some voltage-gated channels on your sarcolemma and even on your, where else could we put some voltage-gated channels? On our T-tubule. So we said in the presence of acetylcholine, the muscle cell will generate an end-plate potential, and then that will activate the production of an action potential at the rest of the sarcolemma and down our transverse tubules. That action potential affects the permeability of the
Muscle cell.
we're in the muscle cell. The action potential of the muscle cell affects the permeability of the restate that. Affects the permeability of the sarcoplasmic reticulum. So when the sarcoplasmic reticulum responds to the action potential, it releases calcium out into the cytosol and binds to troponin of our at least one person's on it. These events are called excitation, contraction, coupling, because acetylcholine doesn't cause the spontaneous contraction in the muscle cell. We've got to couple. What does that mean? It does mean two, so what else was another way? If we're going to couple things. To put them together. I've got to link or join excitation with contraction. So the cell has to have the mechanism to convert this voltage change into a mechanical stimulus or response, really, in the muscle cell. So all of these events, grading up to threshold, generating an action potential, conducting it down the D tubules, leading to the release of calcium, is collectively called excitation, contraction, coupling. Including the excitation and linking it. We haven't got the contraction yet, so we've got to have all these events first. They're called what? Excitation, contraction, coupling. It's all the events that lead up to the release of calcium from the SRs, the sarcoplasmic reticulum. Once calcium's available, then it can bind to troponin of the troponins on our thin filaments. Once calcium's available, what happens? Troponin blocks, very good, the binding sites. And now myosin and actin can bind. Still though, we haven't seen any what? They're just touching, but we still need to do what? Contract. We need to shorten the distance between the Z lines. Those events we'll cover in just a moment, called the cross bridge cycle. We're going to do this cross bridge cycle where these myosin heads get energized and ratchet and yank the thin filaments in, one actin molecule at a time. You can get to contraction. So we'll come back. There's a very sequential series of events during the cross-bridge cycle we're going to cover in a moment. But right now, you say cross-bridge cycle takes place. When, what's available? Calcium from the SR in response to an action potential, which was initiated when there was first an in-plate potential due to the influx of which one? Sodium through our nicotinic receptors because of the presence of acetylcholine from a somatic nerve. Once a muscle contracts, does it need to stay contracted?
No.
No, it needs to also relax. What do you think our muscle cells need to do to favor relaxation? Get rid of the calcium. Don't lose it forever, but do what with it? Store it in the SR. Just put it right back into the sarcoplasmic reticulum. So how do I want to get it in there? If it flowed naturally or spontaneously out, how am I going to get it back in? You got to pump it back in. Active transport it back into the sarcoplasmic reticulum. So we became introduced into the events of the excitation-contraction coupling. In last week's lab, that last question on your pre-lab, it turned your attention right to this image and kind of walked you through it. So having some exposure, maybe having had the quiz or taking the quiz this week, or today, excuse me, what questions do you have about excitation-contraction coupling before we turn our attention to the cross-bridge cycle? Again, you can make reference to figure 12.8. And also those videos that I showed in lab, right? There's a single, I showed the excitation contraction coupling video. Where do you find those at? Mastering, AMP, in the what? AP Flicks in the study area. So go to the study area and then pull up the AP Flicks. There are 3 videos. One is excitation. The other is the contraction coupling. The third is actual contraction. And so it breaks into the 3 events. Let's turn our attention now to the cross-bridge cycle and look at what happens there. During the cross-bridge cycle, it's outlined in figure 12.7. It's a sequential series of events that's very well characterized, and so that's why we get to pick it apart like this. In essence, when calcium is available, a muscle cell is not going to just spontaneously contract if there is no ATP available. So yes, calcium is important for the binding, but we need ATP to power the flicking motion of the myosin cross-bridge, or we talk about the myosin head. And so if we start over here on the, looks like, let's see what's going to start. It's a cycle, so we can kind of start wherever we want. We'll start over here, what's labeled as panel 1, where, what's available? Calcium's available, it's binding to troponin and pulling back tropomyosin and exposing our binding sites. What is already, so your myosin cross-bridge or your head is bound to actin. You see they're touching. But also you see that ATP has been here and it's already been hydrolyzed or split. But notice when it's split, it's the precursors or the products are still there. ADP is still there and inorganic phosphate is still there. You notice how they're still in place. After the binding and ATP is split, our inorganic phosphate cleaves off first. Okay. The sequence is known and well characterized. So first, inorganic phosphate is removed. And that kind of activates the myosin head and allows it to flick, if you will, and change its status. Notice in panel 1 versus panel 2 how it's more relaxed, and then when the inorganic phosphate is transferred off, it flicks. It's bound to actin, so it's going to bring an actin molecule forward however the distance is that myosin head change. Does that make sense at all? You see how it moved inward?
So why doesn't it move till the phosphate's removed?
Because the phosphate is transferring its potential energy. So this is split fast. And so ATP split, inorganic phosphate comes off, and then it activates. So it's not like waiting 2 minutes like what we're doing. It's before we can even think about it. It's happened. And so it's sequential. But a moment later, however long that is, a picosecond or however the difference is, as soon as inorganic phosphate's removed, then ADP is going to be removed. But it's not simultaneous, basically. That inward flicking is called the power stroke. As inorganic phosphate is removed, that changing of the myosin cross-bridge status is called the power stroke. Now what should we do, though? Got to get ready to release it, right? And so we first need to get rid of what? The ADP is going to be released. Notice that when ADP is released, the myosin cross-bridge is still bound. So ADP is off, the thin and thick filaments, correct? The myosin cross-bridge is still attached to actin. So what has to happen is the thick filament or the myosin cross-bridge has to detach. The only way that it's going to detach is the addition of a new ATP molecule. So a new ATP molecule will cause the myosin cross-bridge to detach. And then if calcium is still available, we can reactivate the myosin cross-bridge and then cycle through again. Let's think about a real scenario where a skeletal muscle is contracting, calcium's available, thin and thick filament are touching, but there is no ATP because you have fatigued yourself so much. You've exercised so much, and you're trying to keep pushing yourself, and your ATP levels go down. What's going to happen to your skeletal muscles? They won't be able to relax. Okay? And so they're going to stay, the calcium's still high in the cytosol, the binding sites are still exposed, they adhere to one another, they bind one another, but if there's no ATP, they cannot detach one another. So hopefully you don't ever get into a scenario where you work out that hard. However, this is really prominent if you work with people who have recently deceased. What happens? Rigor mortis. The muscles exhibit rigor. As the cells die, your cells cannot maintain gradients because the pumps can't pump because they don't have any ATP. So calcium's going to flow where? Out of the sarcoplasmic reticulum into the cytosol, bind to troponin, pull back tropomyosin. Thin and thick filaments they won't contract, they'll just bind. This will exert rigor. They won't shorten necessarily, they'll just stick to one another, so the muscles will have a lot of rigidity or tone to them. They will only detach if there was more ATP.
Is that what happens when someone like cramps? Like a muscle cramp? Is that what's happening?
Muscle cramps are generally caused, they could be due to a low amount of ATP, which is correlated to just, so if you're exercising and you're fatiguing yourself, your oxygen levels are low, and pyruvic acid can't go into the mitochondria. Okay, so you got fermentation, so your relative ATP levels are declined. So that could be a reason. Generally, cramps are due to an ionic imbalance, though. when you have low ATP, your just ability to generate, you fatigue, you feel weak, but probably not so much contraction, it's just that weak, fatigue, burning feeling, more than like a sustained contraction. So these